past couple of years, you may have heard a lot about genome editing in the news, at school, or from other people. But what is CRISPR-Cas9? Before that, we need to understand gene modification. Gene modification involves making changes to someone or something's DNA. These changes could have the outcome of enhancing certain traits, preventing genetic diseases, and much more. Cool, right? Humans have already experimented with gene modification in many foods. Let's take, for example, the Green Revolution. In the 1950s and 60s, agronomist Norman Ernest Borlaug found a way to genetically alter seeds so that their plants would be bigger and bear more fruit. The increase in plant size would allow more people to be fed. This was done using other gene editing techniques that weren't as efficient as the CRISPR-Cas9 technique. Currently, scientists are focused on using gene modification to prevent genetic diseases, such as cystic fibrosis, hemophilia, and sickle cell disease. This is where CRISPR comes into play.
You might also be wondering, what does CRISPR-Cas9 even stand for? Well, CRISPR stands for cluster regularly interspaced short palindromic repeats, and the Cas9 mean that it's associated with the Cas9 protein. How does this happen? It all begins with bacteria. CRISPR-Cas9 come from E. coli. Upon exposure to viral DNA, pieces of that DNA gets integrated into bacteria-owned DNA as repeated DNA segments, which is known as CRISPR. Then comes CRISPR RNA that acts as a guide to the matching sequence part of the DNA. The cRNA in the Cas9 protein creates the complex system that finds a matching viral DNA sequence to cut and then uses it to create a new sequence of DNA, hence allowing the bacteria to have its own full collections of protection. But what if we can use this system on humans or other living organisms?
We can thank Emmanuelle Charpentier and Jennifer Doudna, who thought of this back in 2012 when they created the CRISPR-Cas9 technique. They won the Nobel Prize in Chemistry for their discovery and the tracer RNA.
Many people have concerns about human gene modification and the CRISPR-Cas9 technique. Although CRISPR-Cas9 has a high degree of accuracy, it can still make mistakes. Would CRISPR create unrealistic expectations on desirable traits in the human population? Would the super wealthy have more access to CRISPR? The patenting of CRISPR techniques will make it less accessible for the general public. Think back to the Green Revolution. While crop quantity increased, it lowered crop quality and caused a decrease in biodiversity. Of course, CRISPR-Cas9 has its benefits too, as clinical trials with humans and mice returned with positive results.
Created using Powtoon.